Hello, everyone. We're waiting our expert, so just a second, please. We'll start soon. Oh, I think we can start. So again, welcome to the third International League of the BioTournament. And we start the second. Just a second. We start the second circle of scientific fight. So first of all, let's start with introducing everyone. So my name is Tatiana, and I'm your chairperson. There is Alina Frolova, and she is a member of counting committee. She will count your scores, as you understand. And then let's check our experts. I just introduce you, and you may say hello, or that you're there, something like that. So, Berezhnov Alexey, PhD, Institute of Cell Biophysics, RAS.
Hi, everyone.
Okay. Zayas Evgeny, Research Engineer, Laboratory of Biopharmaceutical Technologies, Chemakin Avchennikov Institute of Bioorganic Chemistry.
Hello.
Okay. Kvitkina Anna, PhD, Institute of Physical, Chemical, and Biological Problems of Soil Science. Anna, you should turn on your microphone, please.
Hello.
Okay,
Hello.
good. You can turn off it now. Okay, thank you. Aladin Danila, researcher, Institute of Basic Biological Problems, RAS.
Good day, colleagues. I'm glad to see you.
Okay, Tutokina Maria, senior research scientist, Skoltech.
Hi.
Stavnov Stanislav, head of the Laboratory of Public Health Indicators Analysis and Health Digitalization, Moscow Institute of Physics and Technology. Are you here, Stanislav? I don't hear you.
Okay. Hi there.
Okay. I see you. I hear you. It's cool. And Bokan Bella, Institute of Genetics at Molecular and Cellular Biology, Strasbourg.
Hello.
Okay, cool. Everyone is there. So now we'll check our teams. There. Thanks for difficult names of your teams. First of all, Frantic Multicellular. Are you here?
Yes, we are.
Okay, cool, guys. Bacterian Rhapsody.
Here.
Okay, and Vichikova.
They're here.
Okay, cool. So let's go to the Capitan's quiz. And I think you know how it will be. I just repeat it again. So you will send your answer in chat with me, not for everyone, just with me. And we will have just 20 seconds for this answer. And you, of course, you should type it in English. So I just show my screen. And start. Time's up. It looks like I don't have your answer. Is it true, guys?
Definitely.
Okay. Do you need more time or do you need another task?
Honestly, we don't have the
Another task.
Okay. Okay, so let's go to another task. You will have 20 seconds as always, and it's just number. You should just send me a number, okay? I think you can handle it. So be ready. And just a second, please. I tried to do it again. Start. So the right answer is 5. So Vichikova is a winner. And the Capitan can choose all of positions for teams. So let's start. Just a second. Okay, yep.
Okay, let me think for.
Okay, you have 30 seconds for it to take your decision. Okay, dear Captain, I need to know your decision. What kind of line do you want? Do you prefer?
Yes, we want to be the first column, and we want to report the task first. And then
Just a second. Could you repeat, please? What kind of role do you want to play first?
a reporter.
A reporter. Okay, I just texted. Sorry. Okay.
Okay, and the third would be the team Frantic Multicellular.
Okay, and the second
And the
is
second Bacterian Rhapsody.
so. At the first challenge, Bacterial Rhapsody is an opponent team, and Vichikova is a reporter. So, the opponent team, which kind of task do you want to challenge the reporter?
Give us a sec, we need to figure it out. 1 moment.
Okay, 20 seconds.
Okay, we choose number 5.
Okay. So the reporter who will be?
The reporter will be Daria.
Hello, it's me.
Okay, who will be the opponent?
The opponent will be Albert Kolsov.
And who will be the reviewer?
Alina will be the reviewer.
Okay, cool. So dear reporter, you can start to prepare for your presentation.
Good afternoon, jurors and colleagues. My name is Daria, and I represent Vichukova team for the task number 5. The main point of the task is to suggest an alternative method of genetic collection of perennial crops for their further application and selection. Let's start with different conservation techniques. The first method is in situ one, which proposes the biomaterial conservation in their natural habitats, such as biosphere reserves and other protected areas. But in situ methods are space and time consumption. Another type is ex situ conservation, collection outside the natural habitats. The most used technique is seed banking, but for that, we're able to use just special group of seeds. Thus, we try to improve cryopreservation conditions for better cell regeneration after the liquid nitrogen storage. The first problem should be solved is what to use. The orthodox seeds could be dried and stored in low and even liquid nitrogen temperature. Beside them, there also exists the recalcitrant, the opposite of the previous one. For example, most tropical fruits are recalcitrant seeds. Moreover, vegetatively propagated plants should be collected as well. Thus, we suggest to use meristem and callus for cryopreservation. The great advantage of meristem is low water saturation according to cytological morphology. But meristem compared to callus is less survival after the regeneration. The scheme of our solution is on this slide. We also suggest is on this slide. We also suggest to collect the pollen if it will be possible. The regeneration tissue could be used in lab work or in industry. We also suggest to collect. The next application is cloning the parent organism. And the addition, the pollination by regenerated pollen leads to biodiversity. In our research, we focused on these problems, especially on preservation one. As you remember from the previous slides, the huge challenge in cryopreservation is cell death, which occurs due to ice formation and degradation. Cell preparation before the preservation and the formation of glassy ice may avoid these problems. There are special plant vitrification solutions which consist of internal and external cryoprotectants. The external one, according to their penetration ability, has cytotoxicity, but the application of external without internal is meaningless. So for better result, the mixture of both agents is used. The most popular are plant vitrification solution 2 and plant vitrification solution 3. The difference is that PVS2 contains DMCO and ethylene glycol, which increases the toxicity of the solution. But according to them, the prevention of ice formation is better in the second one than the third one. The possible solution is to decrease DMCO, ethylene glycol concentration, and increase glycerol. Such PVS shows on this slide. Before the PVS, cells should be treated by less concentration solution, which called loading solution. Here is the concentration of different substances which include this solution. Another function of loading solution is degradation and osmoprotection. Instead of PVS2, we can use PVS, which is less cytotoxical, which has less cytotoxicity. In attempt to achieve it, authors saturated the ELS with proline, which has cryoprotectant property. In constant of every PVS, LSP isn't cytotoxical. But researchers preserved cells in -80 for 8 hours. Thus, we suggest to use PVS after LSP treatment for liquid nitrogen storage. Before all preparation, cells should be encapsulated for avoiding extra degradation. The sodium alginate were used for that process. The condition of treatment depends on plant species. After the normal regeneration, cells divided and tore the shell. Finally, we got to the last question in the cryopreservation, is how to collect. We propose to use cryoplates. As you may see on the slide, each well in the plate is dedicated to its biomaterial for avoiding space consumption and losses. Actually, classical cryotubes are also applied for a rainy day or if the special biomaterial is needed. Well, let's remind this scheme and complete cryopreservation method. Firstly, cells were encapsulated, transported on the plate, and then treated in PVS or LSP condition. If the cryotubes prepared, cells firstly were treated and then transported in the tubes. Next, the -30 freezing took place for 4 or 6 hours and moved in liquid nitrogen storage. Except the preservation methods were also suggest the further application of biomaterials. For sex determination, hormone therapy was used. After the pollination, a new genetically recombinant organism grew up. We also found the possible application in industry in the near future. Research claimed that the deposition of colors on the gel matrix and further lignification initiation leads to development of woody products. Thanks for your attention.
Okay, thank you. So, dear opponent, do you have any questions?
No, thank you. We need a couple of minutes to prepare.
Okay, so let's start. Okay, time's up. Dear opponent, are you ready?
Yes, I'm ready.
You can start.
So thank you for the wonderful and comprehensive report. The speaker was able to deeply consider the problem and focus on the necessary aspects. However, you may have problems with a myelostem. Science, it may not begin to differentiate and into different tissues. Also, what is the probability that the seeds will not collapse under the influence of low temperature? And what will happen after 6 hours of freezing? And can you please focus on hormone therapy? You said little about here. Thank you.
Okay, dear opponent, now you have time for your opponent's speech. So you should just speak about disadvantages and advantages of this decision. You can ask question while polemic. So maybe you should just give us your speech and then go to polemic. Or you want to start from the polemic part and just skip your opponent's speech.
Just keep this.
But actually it is your scores. Because without your polemic speech, you lost it, like the half of it. Do you understand it?
Yes, okay.
So, are we going to polemic or not?
Yes, we're going to polemic.
Okay, it's your decision. Just a second, please. You can start.
May I start, or the opponent should start for the first?
As you wish.
The question
I
is,
start because you have already asked me some questions and I can answer them. You said that meristem want differentiated after the regeneration, but all articles and all resources that I have read was said that it's that meristem differentiate and it's going very well and very good. And mostly the meristem from the shoot tip was used and all was good. The second one you said about seeds, which can be destroyed under the low temperature for 6 hours. But for this prevention, we use special solutions which avoid this problem. The third one, you said to focus on the hormone therapy, but I don't clearly understand what you mean. I can show the slide if you want, and you can ask me more detailed question. Here it is. Like we add hormones like cytokinin or ethylene to the plants, which stimulate female organs, and then add pollen. And after that, after the pollination, we have the new organism, which leads to biodiversity.
Yes, this is the point what I want to ask. Thank you.
If that's all you want to ask, we can try to improve my research, my solution, our solution team.
And why you want to test for your seeds for 6 hours? Why 6 hours?
Actually, it's enough 4 hours for. Actually, it was really cruel to put seeds immediately to the liquid nitrogen because it's very huge temperature difference between plus 20, for example, and minus 196. So we need the middle point, and this middle point is minus 30.
Yes, I understand. Thank you.
Thank you for your questions.
Thank you.
So, as I understand, we have finished with polemic part. So it's time for preparations reviewer. You have just 1 minute.
Me? Yes. I didn't hear that. You.
Yes, dear reviewer, you have 1 minute for preparation.
Thank you.
Okay, time's up. I hope you're ready. Before you start your reviewer speech, please just stay alone before your camera, because now it is your own work individually without supporting of your team.
Okay, I'm alone. Now,
You can start.
first start of, my name is Alina and I'm represented team Frantic Multicellular, and I want to say about Harry Potter. Thank you for your presentation. It was very great. The presentation was colorful and clear. It also have a number of slides. It was helpful to understand it, and it's full of pictures. As I understood, you did it by yourself. It's pretty nice. It was understandable because of the shapes. I don't know how to say it correctly. And let's speak about your model. As I see, the model is well done, but there are some problems with some questions. We will talk about them later. And I think the reporter didn't say about what did we do with the plants who exist in the Manitian flowers and Diosenias plants. That maybe I don't speak well, and these words can be a little bit different. It's great that the reporter said about the problems of crystallization, as I well understood, and have a good solution for frozening the myasthenic cells. And let's talk about opponent. He started with the questions, but the opponent should take a monologue of his speech, and it's good that he had the same question as I had, but in the polemics they wasn't fully discussed. He asked a question, but I think he didn't understood as well why he had some these questions. And for me, the polemic wasn't very good. It didn't make the solution better, and the polemics should make the solution deeper and more successful, maybe. And as a conclusion, the solution for Manitian flowers won't work, because for these plants we need both and hormones. So how could you make them grow as a normal plant? I didn't heard this in your presentation, but the good thing is that solution is great for plants who grow a long time, and for dioecious plants who live in two houses. In Russian, we say like this. That's all for my review. That's all. Thank you.
Thank you. Now we have 1 minute for conclusion from the reporter.
Excuse me. Thanks for your review and for your oppose. And I want to add that I didn't think about Manitian plants, but I think that my method can be widely spread in all kind of plants. We just can represent the meristem or the callus, and that will be work. Yes. Well, thank you. And I was ready to improve my solution with opening, but unfortunately, we couldn't do that. Again, thank you.
Thank you, participants. Now it's time for questions from experts. Dear experts, do you have it? Dear experts, do you have any questions?
Yeah, I have the question for the reporter, the first and the main. Maybe I lost, and I don't know. Where is your list of the literature, and what the percent of this work are your own creation, but not from the literature?
We didn't done the list of the references because we have them at the end of the slides. Give me a second. Like, for example, this references is here, and we have all the references under the slides where we use them. And actually, which we've done in this solution is to combine all methods and all techniques and improve the type of storage of collection, the technique in general.
For what time you're expected that your method, you can collect these, I don't know, plants material, something like this, for what time? Like for a year, 2 years, 10 years, I don't know.
Actually, we can prove it only in lab work, and we can't do that in the thought experiment.
Yeah, I understand, but your expectation?
Well, I think we can do that. We can collect them for a year, and then we need to check every year or every half a year, like how it looks like. It's mostly depends on different things. It depends on the species of the plant and depends on the special solutions which can be added, and different compounds which can be added in the loaded solution or in the vitrification solution.
Okay, maybe I lost. You expect some liquids or something like this for pollen? Because as I know, you can collect it only for the 2 years. Yeah.
Yeah, I see. But actually, as I've read the article, there, the common solution for all types of tissues, and the one difference is maybe concentration or the order of the addition of different solutions, or a combination of these solutions. Like we can use only LSP or LSP, and after that PVS or just PVS. It depends on each type of tissue.
Okay.
Thank you for your question.
Okay, any other questions? Okay, as I understand, our experts don't have questions. So thank you for all of the participants for their jobs. Now it's time for expert just to make a decision about their scores. You have 1 minute. Okay, dear experts, I hope you are ready. So let's start. Mr. Birjnov, please give me your scores for the reporter, the opponent, and the reviewer.
Okay, I put 8 points to the speaker, to the opponent 6, and 4 points to reviewer.
Thank you. Mr. Zayats, please.
Yes, to the reporter 9 points, to the opponent 3, and to the reviewer 4.
Thank you. Ms. Kvitkina, please. Please turn on your mic. Okay, yep.
Okay, I put 9 points. Okay.
The reporter.
To the reporter, yes, 6 points to the opponent and 4 points to the reviewer. Thank you.
Okay, thank you. Mr. Aladdin, your scores, please.
Reporter 7 points, opponent 5 points, and reviewer 3 points.
Okay, thank you. Ms. Tutukina, please.
Reporter 8, opponent 3, and reviewer 4.
Okay, thank you. Mr. Ostavnov, please.
7, 3, and 4.
Okay, thank you. And Ms. Bokan, please.
To review, to reporter 7, to opponent 3, and reviewer 5.
Okay, thank you for scores. Now we are moving to the next challenge. So,
excuse me, I would like to give one minor commentary to the reporter and generally to other teams about the list of literature. It's good that you include literature references in your slides, but I think it would be better that if you duplicate those on an extra slide so that there wouldn't be any extra unnecessary questions. That's all.
okay, thank you.
Thank you for your advice.
Okay, so I think we are ready to start the new challenge. And the opponent is frantic multicellular, and the reporter is Bacteria Rhapsody. So, dear opponent, what kind of task do you want to challenge the reporter?
Give us some time, please.
Okay, you have 20 seconds.
Okay, we will choose the question number 6.
Okay, it is possible, yes?
It seems like that.
Okay, so who will be the reporter?
Reporter will be Kovaleva Alexandra.
Okay, who will be the opponent?
Alina will be the opponent.
Could you please repeat the name?
Alina.
And the last name, please?
Amina.
Okay, thank you. And the reviewer?
The reviewer will be Victoria Konnikova.
Okay, thanks. So, the reporter, you can start to prepare for a presentation.
Okay, 1 moment, I need to prepare it. Okay, here we are, and 1 moment, I need to make it a little bit bigger than that. And can you see it?
Yes, we can. Thank you.
Okay, here we are. So a bit nervous for me, but thank you for your attention and thank you that you came to listen to me. So here we have the task. This is the film supremacy. So what's the problem? We know that there are eukaryotes and they tend to create some blankets. We call these blankets biofilms, and these blankets are very successful in protection of these things. And the largest problem that we cannot dispose of these little prokaryotes just because of biofilms. And we call it EPS. This is extracellular polymeric substances. And these are the things which create the problems. And I have few decisions, I know, but I think my decision, the final decision, will be to combine them. So below you can see the link about my information of biofilms, and this picture is from this article. So let's start. Talking about complications, the main problem of biofilms is impossibility to get rid of them once and for all. So we know that some staphylococcus they are inside the eukaryotic cells, but still we have some biofilms and microorganisms are well protected because of them. Antibiotics cannot dispose of bacteria. They can kill the things which are inside, but not outside. So it causes the reason of appearing over and over again. Weakened immunity ignores pathogens. It happens because existence of harmful biofilms is a result of immunity fail. So we can find these biofilms everywhere, even on our teeth, but usually they cause vaginosis and other unpleasant things. For example, they can harm our lungs. So what do we do? The first solution is quite easy, and I know that one team had something similar. Even if a bacteria is covered, it is still a bacteria and it is weak against bacteriophages. So there are many types of these specific viruses, and I'm sure there are exact viruses which attack staphylococci, but I know that it cannot be final because bacteria are clever enough to create a special immunity, and I know that European tradition is against the bacteriophages just because there is the thought that they can be harmful. But still, we can use them, and this is quite common for our country, for Russia and Ukraine and Kazakhstan and other things. So what do we do? To succeed creation of special ointments, inhalation substances, and syringes are necessary. These ones should contain acetyl-lysetine, stomach chemical enzymes, or more specific enzymes. So talking about ointment, we're about to create the ointment with the modified amino acid and special enzymes. And these enzymes are about to destroy the biofilm and to make the little microorganisms uncovered. And when they are uncovered, they are an easy target for our bacteriophages. But talking about stomach typical enzymes, there are a lot of their kinds, and honestly, I'm sure we can choose something for each type of pH, so that will be good for us. But this is not enough, I know. And I have the second way. Way 2 also contains contamination of bacteria with bioparticles, but this time we don't have the bacteriophages. This time we have something like plasmids. So talking about the structure of operons, we know that there are some special pieces of their genetic information, and this is the piece which creates the distortion for creation of biofilms. And in theory, if we can create the plasmid which contains this little piece, we can stop the creation of biofilms eventually. So if plasmid produces enough regulation mass, it eventually will block the synthesis of polysaccharides, and it causes the vulnerable state of our prokaryotes, our staphylococci, and different things. And here you can see the gene which is responsible for creation of biofilms. Well, in theory, I know that there are a lot of them, but this is ATLE, and this is a little link, so you can check it out. And solution number 3 is very simple and very angry, I think, and it is short-term. If we need to remove the biofilm very quickly, the last variant is possible with syringes and other types of treatment where solutions are involved. So idea of creation of hypertonic or hypertonic solution after uncovering of microorganisms. So we do the same manipulation with our enzymes and our modified amino acid, and we just include the special solutions. So we're about to pop them, or maybe just to make them very small enough to not be alive. But I know that this is a weak solution because it doesn't affect the bacteria which are inside. But for that, we have the antibiotics, and I know that one of the things of our task that antibiotics are involved. So this is their task, not the task of the biofilm, creation of hypertonic and other solutions. But I know that all these solutions can be pretty weak, and there is one more way how to stall the microorganisms. If you are problematic in correct treatment, we can stall them and stop them. And we know that some biofilms, they are dual. But how do you think that the idea of creation of trial may be quarter? Things will be quite good. These little microorganisms, they compete to each other and they are real rivals. So if we cannot kill them once and for all, we can just make them a little bit slower while we are meditating about the creation of better way. And this is the source number 5. And sorry, I didn't put it on this slide, but you can see this in the end. And yes, this is the list of literature. To conclude with, I can say that this problem is quite intergrowing, and I have never worked with different cocci and prokaryotes. So if I have more experience, I'd give you something better than that. But for now, that's all. And thank you for your attention.
Okay, thank you for your report. Please, could you just turn on demonstration of your screen again? Maybe somebody need to watch on your presentation or some kind of details. Okay, thanks. That's enough. I think so. Dear opponent, do you have any questions?
No, I have 2 minutes, yes.
Okay, you have 2 minutes for preparation.
I'm ready. May I start?
Yes, of course.
Hello, my name is Alina, and I'm representative of the team Fantic Multicellular.
Just a second, please stay alone because it's your individual job, individual work. Yes, it's just a rule.
Okay, now I'm alone. Thank you for your presentation. It was clear and it was really nice with nice colors. You also have a number of slides and reference list and the links under the pictures that you didn't do by yourself. It's great, no, as I think. And let's talk about your solutions. The first benefit is that you have a lot of different solutions for different problems that can use in different situations. It's very interesting, and I think in Viva it could be successful. Let's talk about the using of viruses. It's not something new, it's already used. It's the minus. And let's talk about plasmids now. You didn't said about how can you transfer a plasmid, or maybe I lose it. How can you transfer a plasmid into the bacteria cells, and how you can save it, because the plasmids consist of a DNA, and they will be destructed, as I understand clearly. And another variant, another question about the plasmids. You didn't say what can we do if bacteria notice that these plasmids are bad, and she can use these plasmids. What solution can be in this variant of the way? And it's really nice that you can mix all your solution in one therapy. So we didn't speak about the therapy in vivo, but maybe we could do it in Polanix, and you will answer to my question that I already noticed, and we can talk as a conclusion. We can say that some specific of your solutions could be better done. So let's talk about what's new in the viruses solution and how you can transfer the plasmid to bacteria cells, and what about the plasmids, how can you save them in vivo, how you can, and as a topic to speak, I want to speak about ORI. ORI sequence of plasmid. You said that we can modify them, and I also know that we can the numbers of copy of plasmid can depends on the ORI sequence of plasmid. So what do you think about it? Let's go to the polemics. May I start? Yes.
Just a second, please. Just need. Okay, yes, you can start.
So the first question, I think we would discuss the plasmids. Yes.
Okay, may I start?
Sorry, I didn't heard you.
May I start? I could explain it.
Yes.
Okay, starting about the first thing with bacteriophages. Okay, it isn't new to use bacteriophages, but it is new to read syringes with enzymes and with acetylcysteine. That's pretty new and pretty fresh because we used a special ointment or maybe a syringe with mixed things. So these aren't the pills. we usually use the pills, usual capsules with these bacteriophages, and they do their stuff. But now we are trying to do something different. So my influence in the destruction of their biofilm by different enzymes, and when the bacteria are vulnerable, we just use our bacteriophages, and in theory it can be really fast. But I know that bacteriophages are not so new for us, and I know that in theory this way is already existing, but that was the first thought that came in my head, so I just offered it just for noting. And yes, I'm saying that this is not the best way to make the problem be nice. And now let's talk about the solution number 2.
Oh, wait a minute, please. Because of your answer, I have another question. Am I right that the specific proteins that can destroy the EPS is inside of the surface of capsid? Yes, or the viruses? Yes or no?
No, not. We're about to add it into the ointment. And how to get it? Well, it could be the simple analogs from our saliva. So we have a big diversity of different enzymes. It's just a task to find them. If I had the lab, I had found it already. So
Okay,
yes.
thank you.
It's not the
It's clear. Understood.
effects. It's the problem to find.
And let's talk about second questions. Question you asked, you started, but I interrupt you. Okay.
Okay. Well, who's me or you?
I can barely hear you. You are muting sometimes. Can you repeat, please?
Okay, who's speaking, me or you?
You are speaking, but
you are asking. Okay, I understand. I remember your question about solution number 2. Can you hear me?
Yes, now it's okay.
So you asked me how to use the plasmids. Okay, it's a little bit brave, I think, but all the operants inside the bacterial chromosome, I know that it's not very correct to call it chromosome, but all these things, they have the special regulatory center, and this center is responsible for spawning the new proteins. If the protein sits on the operator stops working, and this is the main idea. So I want to add the plasmid, which is responsible for creation of this protein. So my plan to overproduce this thing. And how do I want it to spread? Well, we know that all bacteria, they spread and they share their plasmids. So in theory, it's can be acceptable, but I have no experiments, honestly, because as I already said, I've never worked with cocci, and only little amoebas on my decision. But if we overproduce this protein, and if we can to introduce it into the cells, it will spare us of different bacteria films, biofilms, I mean that. And in the end, we can use our antibiotics, and this is the way we are about to solve it.
Okay, as I understood, you use, you don't need to transfer the plasmid into the bacteria cells, yes? Or you should transfer?
Okay, this is the story. We are taking one cocoa, and we need to extract a little piece of this little stopper. I don't know how to call it correctly, but this is regulatory again. It regulates. We need to find it, and we need to extract it. If it is extracted, we need to attach it into the plasmid, and the plasmid can be any. And this modified plasmid with their own regulator.
I understood.
Yes, and we need to
introduce. But how we can transfer this plasmid into the cell culture? Because if we use as I understood, we use the bacteria cell and we transfer the bacteria cell with the plasmid, yes, into the cell culture, yes? So it's clear. Thank you. I think it could be work, maybe.
One more thing, we know that they share plasmids, right? I know that the transfer of genes is a very problematic stuff, but still, we need more experiments. And in the end, if it doesn't work, we can just simply make them to compete. Like in the last slide, well, not last, but before the last, nothing works. Different dual biofilms, they compete, and their metabolism, it's getting lower and lower if different forms are represented. So if even our biothing doesn't spread everywhere, we can just stall them, and the stalling of them is also a good opinion from the beginning.
Okay, thank you. And
okay, dear reporter, you have just 7 seconds. And the opponent, you still have 3+ minutes.
3 minutes to conclude, yes?
No, no, for polemics, you can use it like you wish.
Okay. In the conclusion, I want to say that after the answers of the reporter, I understood the model more clearly, and I think that using the bacteria, the plasmids, using that we want to transfer to the cell culture using another bacteria isn't very successful therapy, because we just make another bacteria to the biofilm, and it doesn't mean that we will share this plasmid. They have F+ plasmids to share plasmids among them from one to others, and it doesn't work as well as to be a treatment as for me. And what else did I wanted to know? Oh, yes, we didn't discuss how we can treat biofilm diseases in vivo. It's a polemic question. And in the question of question number 6, we need to explain the treatment of these diseases.
May I speak?
Yes, certainly.
Our problem just to dispose of biofilms. If biofilms are in there, it's very easy to deal with antibiotics.
Sorry, but your time's up. Dear reporter, your time's up. Dear opponent, you still have 1 minute and 40 seconds.
Okay, and another conclusion that we still have some questions that we didn't discuss, for example, the therapy of biofilm diseases, and even though the reporter's solutions, there are a lot of solutions, you can mix them all to make very successful solution, maybe. And that's all that I can say. I also said 2 another conclusions, so that's all. Thank you.
Okay, thank you. Now, dear reviewer, you have 1 minute for preparation.
Okay, thank you.
Time's up. Are you ready?
Yes, I'm ready. Can you hear me?
Yeah, everything is okay. You can start.
Okay, good afternoon. Thanks for the presenter and for opponent solutions. First of all, I would like to mention the reporter's speech. I would say that the speech and the presentation itself and the pronunciation were really good and were really nice to hear. Thank you very much. In this presentation, we have seen 4 possible solutions to biofilm problems. The first one is the bacteriophage plus matrix disruption enzyme solution. The second one is a bioengineering solution, then solution based on hypertonic solutions. And the fourth one is the ecological is of adding different bacteria. These all are pretty much fundamentally interesting, but I would like to stop on them step by step. First of all, use of bacteriophages and matrix disruption enzymes. I don't think that the novelty of adding matrix disruption enzyme is really useful because many bacteriophages actually have enzymes that can disrupt matrix in them, so it's not really logical. Then hypertonic solution is also has things to consider because it's not really clear how can we selectively put it on a biofilm, and also it might cause necrosis of cells around the biofilm. So there are things to consider. Talking about the negative parts, I haven't really understood how can you plan to combine all these 4 decisions into one and translate it into a medicinal application, because yes, from the fundamental point of view, these all are pretty interesting, but how can we treat patients with all of these 4 decisions? And there's also have been no economical part, and I think some of the decisions, for example, the plasmid one, is kind of complicated. So you need to think about that. And talking about the opponent, there has been a really good introductory speech. Thank you very much. She has mentioned the good and the bad sides of the presentation, and the whole speech was in really positive mood. Thank you very much for that. The polemics was really active. We have heard some of the clarifications of the mechanisms, especially the bioengineering part of the solution. And there also has been some influences made on F+ plasmids, but we haven't heard the transformation of 3 or 4 fundamental things into one medicinal solution. And also, there has been no possible immune response considered, and no antibiotic resistance problem clarified. So there still are things to consider, but to sum up, I would tell that the reporter and the opponent has done their job really well, and the polemics and the presentation were really productive. Thank you very much.
Okay, thank you. Now the reporter have a minute for conclusion. You can start.
Okay, let me complete it. Thanks for your words. Thank you for your attention. I know some ways are problematic. Well, just to explain, well, introducing of enzymes is just a way to make it more efficient. Of course, bacteriophages usually have their own way to be efficient, but of course, it's good if we make it even more efficient. And yes, I know it isn't ideal, but I need more experiments. Talking even about this hypertonic and other solutions, well, it depends on tissue. Not all the time they can create the problems and the necrosis. That's not necessarily. It really depends on the tissue, but it's very difficult to say it in theory because I need experiments. Without experience, I'm blind. So thank you for your attention. Thank you for pointing in my weak spots. That's was really nice. And opponent and the reviewer, that was really cool. I appreciate it.
Thank you. Now it's time for expert questions. Respected members of jury, do you have any questions?
Yes. May I ask a question about the usage of plasmids in vivo? So actually, the usage of plasmids and such bioengineering technique is quite cheap. So from the economical point of view, it's not very expensive and it's quite nice. But how would you use it to the real patients?
Okay, let's imagine that we can introduce it into the biofilm, a small colony of little cocci, and they just spread. I don't know, but maybe it works. Maybe we can create a virus, but it's very dangerous to me to say create a virus because, well, usually it's not very well to say because people are usually against the creating of virus. From the beginning, I wanted to create something which will more efficient of contaminating the bacteria. I want to contaminate bacteria with this little gene, but plasmids are slightly more attractive for me than viruses. From the beginning, I wanted to introduce a virus, but not bacteriophage. I didn't want to destroy them. I wanted to make them less efficient, just to forbid them creating the new biofilm.
The problem is that the usage of any genetical construct is prohibited. Okay, so we cannot use plasmids to any medications for now. So we cannot use genetically modified bacteria. Yeah, and this is the problem. But generally, the idea of modifying the metabolic processes by the repressors is really nice, and I really like it. But think about the usage of ligands, not the plasmids, but of modifying the same enzymes, the work of the same genes, but in the genomes, by using the ligands, which make, for example, your repressor active or inactive, something like this. Yeah, but the idea is really good. Thank you.
Okay, any other questions?
I can ask a question about your extramal way with osmotic stress. Do you think that can lead to death?
I'm not sure, but in theory, if we do it rapidly, maybe. I didn't see the cocci. It's just an idea. I've seen only erythrocytes. If I see the cocci, maybe I created something else. But yes, in theory, if they are not prepared and if it is very fast, if it is a strike, maybe it is acceptable. Maybe not, for instance. So, of course, they won't like it. I mean, bacteria.
You know, usually more evolutionary, more, you know, how young organisms, more complicated, yes? It's not so, how it can be dead is much earlier than ancient organisms like bacteria or viruses.
Okay, but what about not killing them, but weakening them? What if the bacteria have more serious problems, not creating biofilms, but fighting with the pressure?
Okay, thank you.
Thank you too.
So the experts, do you have any questions? Okay, as I say no, so you can start to thinking about your scores and you have 1 minute for it. Okay, the experts, I hope you're ready. And let's start from Mr. Berezhnov. Give me a score,
Thank
please.
you. My scores are 10, 8, and 5 to the report or opponent and reviewer, respectively.
Okay, thank you. Mr. Zayats.
7, 8, and 5.
Okay, thank you. Ms. Kvitkina, please. Ms. Kvitkina, do you hear me?
No, she not in conference. You see.
She had quitted.
Okay. So, Mr. Ladin, give me your score, please.
Oh, for reporter, 7 points. For opponent, 8 points, and 4 points to reviewer.
Okay, thank you. Mr. Tukina.
Reporter 10, opponent 9, and reviewer 5.
Okay, thank you. Mr. Ostavnov, please.
9, 9, 6.
Actually, the maximum point for the reviewer is 5.
5. Okay, sorry.
Okay. And Ms. Bokan, please.
8, 7, 4.
Thank you. Okay, thank you guys for your scores. Let's go to the last challenge. And just a second, I'll show you my screen. So now the report is frantic multicellular and the opponent is Vichikova. So the opponent, which kind of task do you want to hear?
I would like to oppose a frantic multicellular for second task, bitte nicht bitte.
Okay, it is possible, yes, Elena. Okay, so who will be your, okay, dear frantic multicellular, who will be your reporter?
Hello, my name is Radayeva N, and I will be a reporter.
Could you please repeat your name?
Radayeva N. Radayeva.
Okay, thank you. Dear opponent, who will be your opponent?
Darya Sherman will be the opponent.
Okay, and reviewer, who it will be?
It will be Aganesian Yelena.
Okay, thanks. So, just a second. Okay, dear reporter, you can start to prepare for your speech. Okay, dear reporter, are you ready?
Yes, I'm ready.
Okay, you can start.
Hello, everyone. My name is Radayeva N, and I'm a member of Frantic Multicellular team. Today, I want to tell you about second task that's called Beat Niche Bitter. I needed to find the reasons of the bitter taste of protein hydrolysate and find ways to get rid of the bitterness from the soy protein isolate. So the protein molecule has a structure of a globule, the inner part of which consists of hydrophobic or non-polar amino acids, and the outer part of hydrophobic amino acids. During proteolysis, enzymes and water break down the protein into the free amino acids and peptides. Amino acids that have a hydrophobic radical are called non-polar. These amino acids that is bitter. Free non-polar amino acids are the main cause of the bitterness. They contain hydrophobic ends. We can say that's the one of the reasons for the bitter taste of enzymatic protein hydrolysate. It's broad spectrum proteases. Since they completely break down the protein molecule. This releases free amino acids and peptides, including amino acids with hydrophobic ends. These amino acids are bitter. Ways to reduce bitterness on the example to soy protein isolate. First method is use of endoproteinases and exoproteinases. Endoproteinases are proteinases that break peptide bonds within a molecule in contrast to exoproteinases, which break peptide bonds with the terminal exoprotease. It's peptidase that broke the ends of peptides. Then we can cut the protein molecule with endoproteases, and then we can use exoproteases and cut the bitter ends. Then, as a result, we can get a peptide chain without bitter amino acids. And then we can use wide range of proteases and get hydrolysase. Benefits and drawbacks of the method. Its benefits, its quality and product. We can get quality and product, and bitterness is completely absent. Drawbacks. This method absolutely laborious and because it's difficult to find the correct narrow-acting proteinases. Second method is hydrophobic chromatography. In this method, we can use peptide solution, which we pass through the column with a sorbent. Their hydrophobic ends have the ability to interact with hydrophobics absent. The more bitter peptide chains are absorbed by the salt. Absorption increased with increasing salt concentration in the model phase. Therefore, elution is usually performed with gradient of stepwise salt reaction. Then we can use broad spectrum proteases and get ketorase. Benefits of this method is a fast process and economically beneficial. Drawbacks is bitterness remains and more material is used unless than material outlet. The method that I have proposed is a way to remove bitter amino acids of peptide chain. Methods that allow a bitter amino acids to lose their bitter taste. Still in development. Also, using the methods I have proposed for separating hydrophobic amino acids from the breast. It's possible to conduct experiments separately on bitter amino acids. I believe that hydrophobic chromatography is a more effective method on an industrial scale because it's fast and not labor intensive. Well, that's all. Thank you for your attention.
Okay, thank you. So now the opponent, do you have any questions?
No, I haven't got any questions. I'm ready for the preparation yet.
Okay, so I have 2 minutes.
I can start.
Are you ready?
Yes.
Okay, you can start.
My name is Sharman Daria, and I'm the opponent from the Vichchukova team. And firstly, I want to say thanks for the speaker and for her presentation. It was very nice and very well structured. But let's start positively. We noted the problem. We noticed and but we noted the problem of the task.
the reason of the bitterness, but we also the speaker said about two methods and their pros and cons. It was very good and really great advantages of your speech. Actually, you also consider the economically part of the solution, which is also mentioned in the task. But let's begin with the negatively points. There is no solution for your task. You didn't do that. Because your solution, as I understood, is throw away bitterness amino acids, actually hydrophobic acids. But that can't be possible because the most popular sports supplement is BCAA, and it includes valine, isoleucine, and leucine amino acids, which are hydrophobic. And the second, the most popular hydrophobic amino acid is tryptophan, which is also very important in our organism. And we can't just throw away this amino acid from the soy protein while we hydrolyze it. That can't be. The second point which I want to notice is that is poor knowledge of the basic principles of this task and general peptides and biological activity of the amino acids. Can you open the fourth slide if it's possible? Actually, if it's not possible, I remember that you didn't mention natural amino acids in your column of the bitterness amino acids. Yes, stop, false. Yeah, here it is. There is a butyl serum, as I understand, but what about valine, leucine, alanine, tryptophan, and others hydrophobic acids? And I have one more question on the 11 slides, but that we'll discuss in the polemic. And I really, I would like to help you to improve your solution because we have some positive notes in your already prepared solution, and we just need to improve it a little bit, and that will be a great solution for this task. Thank you very much. We can start polemic if you want, and you can answer my question.
Okay, just a second. You can start polemic, but I need to prepare for it. Okay, so you can start.
Thank you for your question. How I know hydrophobic radicals.
Sorry, can you speak more loud? I don't hear you, please.
How I know hydrophobic radicals in amino acids are non-polar, and it's only 8 amino acids, no?
Actually, I don't know the number of them of the hydrophobic amino acids, but they are all essential. We can't just throw it away from our hydrolysates. But let's discuss the 11. Can you turn on the 11 slide of your presentation? Oh, it's the second method. Sorry for that. Second method. Stop here. Stop here. Like we have peptides which are blue and they are quite short compared to the whole protein. How you are going to hydrolyze it? By endopeptidase or exopeptidase, as I said. Yeah, as I understand.
It's two different methods.
Yeah, I see, but if you see there is a short peptide compared to the whole protein, like imagine the whole protein has a lot of amino acids inside it, like started from the 500s and over, and you here have just 7. Like you want to chromatograph.
I understand.
You just like want to use chromatography for the whole protein because it's have a lot of hydrophobic acids and it's just stay in the column and you wouldn't get the yield. Like firstly, you need to cut this.
Of course, we can do this, for example, endopeptidase.
I see.
Just cut and when we used hydrophobic chromatography, we put it must be not with it, it must be раствор.
Okay, let's help you. Like my my aim is to improve your solution with you, and let's imagine that we aren't going to throw out hydrophobic acids, and we need to stay them in our hydrolysates. How we can do that? Maybe we can hide them inside the hydrophilic acids, amino acids. Like we can make 3 peptides, which include hydrophilic acids, hydrophobic acids, and hydrophilic acids. And perhaps it would be less bitterness than the just hydrophobic acids, or if it will be at the end of the dipeptide of the.
Hydrophobic chromatography, it's a method when you get a less bitter amino acids hydrolysate. When you get a not clear solution.
Well, I see, but let's try not to separate hydrophobic and hydrophilic acid, because as I have already said, both of these types is useful for the organism. Like, okay, can you turn to the first method and let's continue our improve. Yes. Look, here you got 3 bitter amino acids, 3 hydrophobic amino acids. Maybe it's better to use specialized enzymes which cut between hydrophilic acids, and we can get particle peptides, which includes hydrophilic and hydrophobic amino acids, and which can be less bitterness.
Yes,
bitter.
I understand. Yes, maybe it will work. Yes, I understand what you mean.
Yes, like your first method is actually good, but it's need more detailed information of peptides, of enzymes, and I think that we solve this a little bit solve this problem. We make a conclusion that we can use specific enzymes which cut between hydrophilic acids, amino acids. Okay. I didn't see the economical part of both of these methods. I saw that I saw a few words about that, like it was economically good or economically bad, but actually, you really think so?
It can work. It can be economical, effective. For example, not. For example, second method, because it's faster and it can be.
But well, as we already discussed, the chromatography method is a bad method because in this method we separate hydrophobic and hydrophilic amino acids, and that couldn't be the solution for this problem. Let's discuss the first method about peptides, about enzymes. Like, okay, I can ask you a question. Like, for example, you are a director of the factory and you want to separate and you want to use enzymes for cleavage the amino acid for cleavage proteins. How are you going to get these enzymes? You are going to buy it in the shop, for example, yeah, or you are going to produce it by yourself by microorganisms like E. coli and get the recombinant protein.
By myself, because it will be cleaner. It's
well, actually, yeah, it
Just
will
a second,
be.
please. Just a second, please. Dear opponent, you have just 13 minutes, 13 seconds. Dear reporter, you have more than 2 minutes.
Well, I just want to say that the application of recombinant protein is better because it's less price for you. Like you have bacteria which produce a lot of proteins, and that is.
Time's up, dear opponent. So, dear reporter, you still have time. You can say something as you wish.
I believe that solution, what I think so. We can start to listen review. I think we can start to listen a reviewer.
Okay. Okay, so dear reviewer, you can start to prepare. You have 1 minute.
Oh, only one? Okay. I want to say thank you.
Just a second, please. For preparation, you have 1 minute.
I don't have to prepare. I'm ready.
Okay, so you have 5 minutes. Let's start.
It's cool. So I want to say thank you to the speaker and to the opponent for the excellent polemic. I would like to point out a very clear presentation. Really, everything was clear to me and my team. The diagrams, the drawings, the schemes, it was really perfectly matched. I want to appreciate that several methods have been proposed to solve the problem, and what more importantly, the pros and cons of each method were spelled out. It's really cool for solve the problem. It's help us to understand what method, how will work. This method, how it will done. The summaries all have been said and helps to understand the essence of the method. By the way, for the opponent's classes, I would like to note the correctly selected questions about the method of solving problems. The emphasis was placed on the really unclearable moments, and the polemic helped to understand a lot. So thank you so much.
Okay, thank you. Now we have time for conclusion word of reporter. Dear reporter, do you need it or not?
Yes, yes.
Okay, so let's start.
The method that I have proposed, it's a way to remove bitter amino acids from the protein molecule. Methods that allow the bitter amino acids themselves to lose a bitter taste. I think it's still in development. it's very interesting. Thank you for your questions.
Dear reporter, your time's up. Thank you. And now it's time for questions from experts. The experts, do you have any questions?
Yes, I have a question. Could you please turn to the slide where you list the amino acid radicals that cause bitterness? Back? Yes, it's this. Next slide, please. Next. No. Where is a list of? Yes, this one. On this slide, I can see thiobutyl, actyl, other radicals. Those radicals are not from natural amino acids. So my question is, where do these radicals come from which molecules, and what are you planning to do with these molecules?
I read that these radicals in originals in natural amino acids.
So these are from unnatural amino acids.
Yes.
Mr. Zayas, have you finished with your question?
Yes, yes.
Okay, so maybe any questions from others?
I have a question about scaling of your methods, because your methods about you talking about quite expensive, and in tasks we need to I think get some cheap and in bigger scale proteins. How you think about big big matter, about large scale?
I believe now it's yes, it's really expensive. But if we want to remove bitter all in hydrolysate, I believe it's work will be work.
Because soy protein, it's quite cheap. It's several
Yes.
less than $1, several cents for kilogram or for pound, for example. And well, if we try to do, may make them not bitter, make thousands of dollars per kilogram. It's very expensive. So all our tasks in this BioTournament, it's quite applied tasks, and we always think not only about scientific methods, but about money and scaling of these processes.
I will work on about this question. Thank you.
Okay, thank you.
Okay, thank you. The experts, do you have Any
May I
questions?
ask you one more question?
Okay.
I have a question concerning your first approach. At the end of the reaction, you will have the mixture of proteinases and the product of the reaction. And how do you think, is it easy to separate them? You will need to separate them, or it may arise a new problem.
I believe it's no, I think it's problem how to separate.
How to say, it will raise the cost of your approach in realizing your approach. Okay, thank you.
Okay, and as I understand, we can finish with a question from experts, yes? Okay, so now our experts have time. Preparations, yes, of course. The experts, I hope you're ready. And let's start from Mr. Berezhnov. Your scores, please.
Okay, I'll put 7 to the reporter, 9 to the opponent, and 4 to the reviewer.
Thank you. Mr. Zayats.
I put 4 to the reporter, 9 to the opponent, and 5 to the reviewer.
Thank you. Ms. Kvitkina, please.
My points are 7, 7, and 4.
Okay, thank you. Mr. Aladin, please, your scores.
So to the reporter, 5 points, opponent 7 points, and reviewer 3 points.
Okay, Mr. Tukina, please.
My points are 7 to the reporter, 9 to the opponent, and 4 to the reviewer.
Okay, Mr. Atstavnov, please.
7, 8, 4.
Okay, thank you. And Ms. Bokan, please.
7, 7, 4.
Okay, thank you. So we are finishing with our scientific fight today. Dear participants, we will know the results in section number 2 after they have finished with their fighting. You will get the link on Telegram, so just check it. And good luck. I hope we'll see you next time. Yeah, goodbye.
Thank you.
Goodbye. You have a nice cat.
Thanks. I know.